Remember the food mentioned in Part B of this lecture that needs to get imported into your cells to make ATP? Well, how exactly does that food get into your cells? Recall that each one of your cells is surrounded by a plasma membrane through which all substances entering a cell must pass. Here we discuss how cells import substances such as food and how they export substances such as waste products. Without the ability to import and export substances, your cells would quickly die, and you guessed it, therefore you would quickly die. The plasma membrane is selectively permeable. Selective means choosy, and permeable means getting through. Therefore, the plasma membrane only allows the passage of certain molecules into and out of a cell. Since cell membranes typically function in a watery environment, we're going to examine the characteristics of molecules in fluids. In order to discuss molecules moving into or out of a cell, we need to talk about a few terms. First, concentration, which is the number of molecules in a unit of volume. A gradient is a physical difference between two regions, such as inside or outside of a cell, that would cause molecules to move from one region to another. Examples of gradients include concentration, pressure, and electrical charge. An example is a pressurized airplane cabin. When you take off in an airplane, the cabin is kept pressurized so that the pressure inside is equal to the same pressure as it was on the ground. The atmospheric pressure high up in the sky is too low for you to breathe. Therefore, if the windows are shot out of an airplane while you are in the air, all of the air will rush from higher pressure inside the cabin to the lower pressure outside the cabin, taking everything with it. Take a look at the link below for an explanation as to how this occurs. As exciting as it may be to think about windows getting shot out of an airplane, we are now going to turn our attention towards concentration gradients. A concentration gradient is the difference in concentration between one region and another. An example would be the region inside a cell versus the region outside of a cell. We are going to start discussing different types of transport that cells use to move materials into or out of them. The first set of transport mechanisms includes passive transport. In this case, substances move either into or out of a cell down their concentration gradients, which means from a high to low concentration. This requires no expenditure of energy by the cell. Passive transport includes simple diffusion, osmosis, and facilitated diffusion. During simple diffusion, which is the first type of passive transport, a substance moves from a region of high to low concentration until equal concentrations of the substance in both regions are reached. Consider a simple example of spraying air freshener. If you spray air freshener on one side of the room, it will reach the other side of the room pretty shortly. Why? This is because where you initially spray the air freshener now has a high concentration of the air freshener. Across the room has a low concentration. So via simple diffusion, the air freshener will diffuse from a concentration where it's high, where it was initially sprayed, to a lower concentration across the room. In a cell, it's hydrophobic molecules and a small amount of water that utilize simple diffusion. Due to the hydrophobic fatty acid tails of the phospholipid bilayer, these hydrophobic molecules can diffuse freely across the plasma membrane into the cell. Examples of hydrophobic molecules that utilize simple diffusion would be lipids, oxygen, and carbon dioxide gases. Simple diffusion through a selectively permeable membrane moves a substance from an area of high concentration, in this case from the extracellular fluid outside the cell, down its concentration gradient into the cytoplasm inside the cell. Over time, more and more of the molecules move into the cell until an equal concentration is reached both outside and inside the cell. The second type of passive transport is osmosis, which refers to the diffusion of water across a selectively permeable membrane from a high to low water concentration. In this case, it's the concentration of particles or solutes dissolved in the water that will determine the concentration of the water and therefore which direction the water will flow. We will study osmosis by comparing the cytoplasmic fluid of a red blood cell to the extracellular fluid in which it is placed. Before looking at red blood cells, consider the example here. In osmosis, water always moves from an area of higher water concentration to one of lower concentration. In this diagram, the solute cannot pass through the selectively permeable membrane, but the water can. The left side has less solute and therefore a higher water concentration, while the right side has more solute and therefore a lower water concentration. Thus, the water moves from the left side to the right side to equalize the water concentration on both sides of the selectively permeable membrane. Let's first discuss red blood cells in an isotonic extracellular fluid. This means that red blood cells are placed into a solution that has the same concentration of solute and therefore the same concentration of water as the cytoplasm inside the cell. Iso means equal. In this case, osmosis still takes place, but since there is no concentration gradient for water, there's no net movement into or out of the cell, so the cell stays the same size. Body fluids are isotonic to our cells. For instance, blood is isotonic to the blood cells that reside within it. You can summarize isotonic by stating that the solute is equal to the water, equal inside the cell, equal outside the cell. Take a look at the figure to the right and look at the shape of the red blood cells in an isotonic solution. They are round with little indentations in which hemoglobin sits to carry oxygen through the bloodstream. Now let's take a look at red blood cells in a hypertonic extracellular fluid. Hyper as a prefix refers to a lot of something. In this case, the prefix refers to the solute concentration. Therefore, a hypertonic extracellular fluid is a solution that has more solute and less water than the cytoplasm inside the cell. Because there's less water outside of the cell, this sets up a concentration gradient in which water will flow from a higher concentration inside the cell to a lower concentration outside the cell. Because the net water movement is out of the cell begins to shrivel. Hypertonic conditions in the body include dehydration. This means that your blood does not have enough water, and water flows out of your blood cells to equalize the concentration. We all know that dehydration can be deadly. This is because red blood cells in a hypertonic solution lose water and begin to shrivel, and therefore no longer have the indentation required to carry hemoglobin and therefore oxygen. Take a look at the illustration of red blood cells in a hypertonic solution on the right, and you can see this is the case. We can summarize a hypertonic extracellular by stating high solute, low water outside of the cell. While we all know that too little water can be deadly, did you know that consuming too much water can be equally deadly? This would be a situation in which your blood becomes a hypotonic extracellular fluid to your blood cells. Hypo as a prefix means too little of something. In this case, this solution has less solute and therefore more water than the cytoplasm inside the blood cells. This sets up a concentration gradient in which there's more water outside of the cell. Therefore, the water moves from outside the cell into the cell, so the cell begins to swell and burst. Take a look at the red blood cells in the figure to the right, and you can see them getting bigger, losing their indentation to carry hemoglobin and oxygen, and some of the cells are beginning to burst. When your red blood cells begin to burst, they're no longer available to carry oxygen, and you will not live very long without oxygen. Hypotonic conditions in the body include drinking too much water in too short a period of time, or sitting in a bathtub for a long time. After sitting in a bathtub for a long period of time, you see your fingers as kind of being shriveled, but actually what you are seeing is some of the skin tissue is swelling. The swelled portions are sticking up with indentations in between. We can summarize a hypotonic extracellular fluid as low solute, high water outside of the cell. There have been many cases that have made the news that highlighted people dying after consuming too much fluid. Take a look at the link below to see a girl who unfortunately passed away after being forced to consume large amounts of grape soda and water as punishment. The last type of passive transport that we discuss is facilitated diffusion. Keep in mind that all mechanisms that fall under passive transport move substances from a region of high to low concentration and therefore require no energy from the cell. Facilitate means to help. So in this case, substances move from a region of high to low concentration with the aid of a membrane protein. Hydrophilic or water-loving substances utilize facilitated diffusion to get across the hydrophobic fatty acid tails of the phospholipid bilayer in the plasma membrane. These substances utilize carrier proteins or channels embedded in the membrane to enter or exit the cell. Examples of hydrophilic substances are all water-soluble substances, including ions like sodium and calcium that have charges that would be repelled by the fatty acid tails, along with amino acids and sugars which have charges and/or polar covalent bonds. Hydrophilic substances are able to move down their concentration gradient across the plasma membrane with the aid of carrier proteins. Notice that these proteins span the membrane so that these substances can cross it without making contact with the hydrophobic fatty acid tails in the phospholipid bilayer. Carrier proteins change shape as they move molecules across the membrane. Here we see hydrophilic substances crossing the plasma membrane with the aid of channel proteins, which form tunnels through the phospholipid bilayer. So once again, these substances do not contact the hydrophobic fatty acid tails. Since channels cannot change shape like carrier proteins to accommodate larger molecules, they are typically used for smaller substances such as ions. We have talked about three types of passive transport: simple diffusion, osmosis, and facilitated diffusion. All of these mechanisms of transport move substances from a high to low concentration and require no energy from the cell. Here we talk about active transport. It's called active because the cell needs to work to move substances from a low to high concentration. This is the opposite direction of diffusion. Since this is not what normally happens, this requires the cell to expend energy in the form of ATP. It also requires specialized carrier proteins that can hydrolyze the ATP to release the energy. An example of active transport is nutrient import. After we eat, blood glucose is high, and the glucose starts getting imported into cells. However, once the glucose inside the cell is at an equal concentration to the glucose in the blood, the cell then must use active transport to continue moving more glucose into the cell. Active transport moves substances, in this case ions, across the plasma membrane from an area of low to an area of high concentration. Here, sodium is being moved to the outside of the cell to an area of higher concentration, while potassium is being moved into the cell, also to an area of higher concentration. Notice that the carrier proteins hydrolyze ATP to release energy needed to pump these ions against a concentration gradient. There are two other mechanisms of transport that require energy by the cell to move materials into or out of the cell. These are endo and exocytosis. Endo as a prefix means internal or inside, and cyto means cell. So you can think endo, enter, or entering the cell. In this case, cells utilize energy, along with vesicles, to move fluid droplets or large particles across the membrane into the cell that are too large for active or passive transport. Exo as a prefix means outside, so you can think exit, exo, or exit the cell. In this case, the cell utilizes energy to release substances from the cell, also in vesicles. Often a cell uses exocytosis to dispose of waste products or to secrete substances such as hormones. In endocytosis, the cell membrane pinches in to surround a small fluid droplet with dissolved solute to bring it into the cell in a vesicle. Typically, it's the solute that the cell needs and not the fluid itself, So this would be a way for cells to bring many solute particles in at once. Here we see a cell utilizing endocytosis to bring in a large particle that is too large to utilize active or passive transport. A vacuole is a large vesicle, so in this case the particle is so large that it is brought into the cell in a vacuole rather than in a vesicle. In exocytosis, vesicles containing substances fuse with the plasma membrane. The contents are then released to the exterior of the cell.